Let me start by introducing our two speakers. So we have moved all the way to 10 to the 6. So this is about 10,000 years ago, where we are going to look, 10 to the 5, so that's 10,000 years ago. We are going to look at the development of the brain. We have two speakers with us, and we're going to start with Professor Terrence Sejnowski to actually talk about how brains evolved. Professor Terrence Sejnowski is a pioneer in computational neuroscience, and in this lab he uses both experimental and computational techniques to study the biophysical properties of synapses and neurons and the population dynamics of networks of neurons, with the goal to understand the principles that link the brain to behavior. He received his PhD in physics from Princeton, went on to become an investigator at the Howard Hughes Medical Institute, as a distinguished professor at the University of California at San Diego, and holds a Francis Crick Chair at the Salk Institute. Professor Sejnowski is the president of the Neural Information Processing Systems Foundation, or NIPS, which is one of the premier conferences on machine learning and neural computation. He's a member of the National Academy of Science, Medicine, as well as Engineering, and was instrumental in shaping the BRAIN initiative that was announced by the White House in 2013, and currently serves on the advisory board to the director of NIH, Francis Crick, on the BRAIN initiative. I think with these opening words, I'd just like to invite Professor Terrence Sejnowski to give his presentation.
Okay, it's wonderful to be back. I was here last year for the Future of Science Symposium, organized by the same group. And every time they come back, there seems like there's a new building that's gone up, or a new program that's been started, and it tells you something about how fast this place is growing. And I'm really pleased to be here. So the brain is the most complex device in the known universe, if you don't include the rest of the body, which is even more complicated. As my wife, who's a medical doctor, tells me, the brain is just a part of the body. And you might ask, well, why do we have brains? What good are they for? Trees don't have brains. They don't need them because they don't move. But as soon as you move, then you have to have some way of figuring out where to go and when to go. And there is a very peculiar creature called the sea squirt that proves the point. The sea squirt is a chordate, has a spinal cord. It has a larval stage where it swims around until it finds a perch where it can anchor, and it will never move again. And at that point, it eats its brain. Because, of course, it's superfluous at that point, because it has another lifestyle that it's about to embark on, which is stationary. Now, if you look at the different levels of organization in the brain, from the entire central nervous system all the way down to all the molecular levels, there's literally 10 orders of spatial scale. And when we talk about evolution, there's evolution occurring on every single level, which makes it incredibly difficult to figure out how all of those parts and pieces are interacting and how the interactions give rise to activity in the brain, electrical activity, chemical activity, and ultimately to behavior. Now, this is an obligatory slide, but it has an interesting history. This is Theodosius Dobzhansky, who famously wrote an essay and said that nothing can be understood in biology except in the light of evolution. Now, this is actually an essay that was written to counter creationists who, of course, didn't believe in evolution. However, Dobzhansky was actually deeply religious, and he felt that, in fact, they were compatible. Evolution was compatible with religion. I'd like to modify this a little bit. I'd like to say that nothing can be understood in biology except in the light of DNA. And a whole new area of biology began in 1953 when the famous paper by Watson and Crick in nature announced the structure of DNA, and it changed the way we think about biology. It killed vitalism, which tried to explain what's different about life. It had inherent theory for evolution by virtue of the fact that the DNA could mutate and change over time. And I think that now looking back, now that we can have whole genome sequencing for $1,000 or less, that we're entering a whole new era where every single species is being sequenced. In fact, every individual will be sequenced eventually. And we will have a history of evolution writ in the DNA of every living creature that will allow us to track back down exactly when sequences diverged and how that gave rise to different types of behavior. Now, the complexity here that underlies natural selection really starts at the gene, but as you can see, it involves an incredible sequence of interactions occurring, transcription factors that affect development as cells differentiate. And then, of course, the behavior is a consequence of properties of circuits interacting with the world. And ultimately, there's a loop here that gets closed by survival, which is to say that the diversity of in the genomes of the species really are being controlled by all of these circuits and interactions, which, as you can see, have arrows going back and forth. There's information flowing. You should think of this as an information highway that is producing changes occurring. The environment can actually affect gene regulation, and that can affect the future of the circuit. So when you start with an embryo and you start dividing, you go down a path, and the main way that you differentiate is by turning off genes. And the way you turn off genes is by methylating sequences in the DNA. And for example, you can have one cell that differentiates to become a neuron, another one becomes an epithelial cell, and when they are fully differentiated, they will have a subset of cells, of genes that are the ones that are specific for that particular function. Now, the primary form of methylation at that stage is the cytosine next to a guanine, CG, and they're twins, so that you get pairs like this. And this is true for almost all cells in the body, and there are two exceptions. The first exception turns out to be pluripotent stem cells, where in fact about 25% of all the methylation takes place not in the CG context, but in the CH, which is CACT-CC, non-CPG. And we're going to call that MCH. H stands for anything but G. But when the cell differentiates and becomes mature, all of that disappears, and you're left with pure, more or less pure methylation in the CG context. The other exception, that's number one. Number two is methylation in neurons. And this is unique. Of all the organs, the brain is the only one that has mature cells that have a substantial fraction. In the case of humans, 53% of all the MCH sites are methylated. So this is really something that's special and must have some special function. Now, if you look at the pattern methylation, that's giving us a hint. This is a project that I started with Joe Ecker, who's a colleague of mine at the Salk Institute, who's a plant biologist and is an expert at whole DNA methylome sequencing. So we partnered up and we sequenced both humans and in mice a sequence of points during development. And what you're seeing here is just the percent of the methylated CH. You can see that it starts from 0 at birth. It rises very rapidly and it reaches a peak at about 25 years in comparison to the CG methylation, which is already very high at birth and really maintains that high level throughout. So this is the lifespan. Now, it's interesting that if you plot it on the same graph, the synaptogenesis, the generation of connections between neurons, you can see it rises and parallels that of the CH methylation, and then reaches a plateau here near early adulthood. And the same pattern occurs for mice. In fact, it even parallels it even more closely. So this suggests that the new form of regulation that's found in neurons may have something to do with getting the brain wired. Neurons are going to live for the rest of your life, so these are not turning over except in a very few places, like the hippocampus. And so there's special regulation just for them. And it's special in a couple of different ways. First, unlike CPG regulation, which is carpet bombing, it basically carpet bombs both the coding region and the promoter region and the regulatory regions. Basically, you just want to turn it off. It turns out that the pattern of methylation of the non-CG is extraordinarily precise. And here's we're showing here a particular gene, and each one of these tick marks is a C on the two strands, and each of these is the whether or not it's methylated. And these are two different mice, R1 and R2. Now this is actually two different humans. But just compare the pattern, and you can see that there are a lot of sites that are not methylated, that could be, and the ones that are really at the same age, at the same place, are methylated across a wide range. And the same thing true is mice. So this is really, there has to be a whole system here for figuring out when to methylate a particular site and which site to methylate. We have no idea what that mechanism is. However, we do know that like CPG methylation, this is just a very crude, to show you the frequency of transcripts, mRNA, where this is very high at the bottom and low at the top. If you look at the fetus as advertised, there's not a lot of methylation, but here the methylation is very high when the gene is not being transcribed and very low when it is. So this is behaving like a normal form of methylation. Now I'm going to come back to this later because there's another chapter. Just want to say that it's really Joe who drove all the sequencing, and his postdoc, Ryan Lister, who's now at the University of Western Australia, and Margaret Behrens, who's a senior scientist in my lab, staff scientist, and Iran Bukumel, who was a postdoc in my lab, is now in the faculty at UCSD. So this is just to give you some background on how fast things are going, how quickly we're going to know in great detail exactly how genes are regulated and the consequences of that, and tune in for more. Now, in addition to changes that are occurring, obviously, to make all the different species on the Earth, there are some things, it turns out, that are highly conserved. And the Hox genes are a sequence of genes that are found in, as you can see here, both insects and in vertebrates, and they control the segmentation of the body. There are variants that are slightly different in different species, in different mammals, for example. But what's clear is that each one of them controls the development of a particular segment, and in particular, the anterior segments are going to control the brain. And I think this is remarkable that you could have this same mechanism that is used over and over and over again. It doesn't matter what you're going to look like, and how different is a fly from a human, but underlying that, there's this common mechanism, the set of Hox genes. Now, even the most primitive unicellular organisms had ion channels, bacteria. But these evolved over long stretches of time. And by looking at the sequences, you can actually follow how that happened. So the oldest channel are the potassium channels, and then the newest are the sodium and the calcium channels. And you can see, this is really wonderful. This is transmembrane segments here, and this most primitive only has two, the little cylinders, but that led to the rectifier, KIR, and then the voltage-dependent versions had 6 segments, and that duplicated to 12, and that duplicated again to 24, and that gave rise to the more complex ion channels. And so here is one of the principles we see going on during evolution is that you duplicate a gene, and then you create a new function, and you specialize it. Different cells will have different combinations of channels. And in the case of neurons, there may be dozens of ion channels distributed inhomogeneously in different parts of the dendritic tree and the axon. Another good example are photoreceptors. So photoreceptors come in two flavors. There's the ones that we have, which are ciliary, derived originally from hair cells, and the rhabdomeric type that are found in flies. And these have very different mechanisms in terms of the photoreceptors and the ion channels. When your photoreceptor catches a photon, it hyperpolarizes, it decreases its membrane potential, but in a fly, rhabdomeric, it increases. So they clearly have different diverging mechanisms. But as you can see, over the entire kingdom here, there's a tremendous variability, and these photoreceptors were used over and over again in order to create different types of vision for different animals. And in particular, we have 3 different cones, providing us with 3 different channels for red, green, and blue. And these provided us with the ability to distinguish different types of plants and different types of fruit from the background. And this is a good example where you can see if you only had black and white, you might not know that there was a nice juicy fruit here, which you can distinguish by its color. And actually, it's not just the fruit, it's actually all the different types of green foliage, which also are very prominent. And if you are in a jungle, that turns out to be important too. Now, this is my favorite slide because my favorite structure in the brain is the synapse. There are more synapses, like there are on the order of 100 billion neurons in your brain, and that's a very large number. But you know, if I had $1 for every neuron in your brain, I couldn't cover the U.S. national deficit. But there are 10 to the 15 synapses, right? That's an astronomical number, and they're very highly evolved. But the original synapse, actually going back to yeast, was very simple and primitive. And you can see here, these are all the different, this is a phylogenetic tree, but it's been color-coded for different levels. For example, if we look at the presynaptic mechanisms here for the release of neurotransmitter, we can see here that corresponds to things that are already happening here at the very earliest level. This is a very important function. But as things progress, for example, the sponge added a bunch of genes and proteins that allow the communication to go 2 ways. And in the most recent additions, for example, in the vertebrates, we add the TARPs, and this is something that Sydney's very interested in, and also the NMDA and the AMPA receptors. So this is a structure that has been evolving for literally hundreds of millions of years. And it's been getting more and more specialized until you get to the point where it really not just carries information, but has a history of all the activity. And that arises the plasticity that allows the synapse to change strength and allows you to learn and remember things. When were they first created? And this is, unfortunately, we don't really know what was going on back then. This is a phylogenetic scale going up, and here is the place where we think it happened. It happened somewhere between sponges and jellyfish. So jellyfish have something called a nerve net, which is a loose group of neurons that allows the systematic beating, so the jellyfish kind of floats along. Sponges don't have neurons. I guess they just sit there. But as you go up, new things emerge. For example, you have in very early creatures, insects and nematodes, you have typically a lot of ganglia. And then when you have a central nervous system like the brain, that's when you're at the higher level. And then finally, when you get to the vertebrates, you have the appearance of myelinated axons to make the signals go faster. That's a way for nature to increase the speed of propagation. And then finally, the crowning glory that humans depend on, which is the development of the mammalian neocortex. Now, the original cortex actually can be found in amphibia and in reptiles, only had 3 layers, and it had 1 layer of cell bodies, had a lot of inputs, and also the axon spread very broadly. So that type of cortex is preserved in our brain in the hippocampus, which is for long-term memory, and also in the olfactory cortex. So it was how things started, but when the mammals appeared, suddenly it differentiated into a 6-layer cortex with very specialized types of neurons. We don't actually know how many different types there are. There's pyramidal cells, which constitute 80% of the cortex, and inhibitory cells that constitute 20%, but we know that there are subtypes of each of both pyramidal and inhibitory. The other main the kind of structure that we see in the neocortex that was really probably the key to rapid evolution was the fact that it was topographical. So the visual field, for example, is mapped point by point into the visual cortex. And we'll see that that allows a hierarchy of visual processing centers. Early mammals, for example, mice and rats, they have sideways pointing eyes. They have big olfactory bulbs and very small visual cortex. But if you now go to primates, you discover that the eyes move forward, overlapping, allowing stereoscopic vision, binocular depth perception, so that you can more accurately manipulate things with your fingers. And the visual system correspondingly increases in both the number of areas in different colors and also the amount of volume. And you can see that in primates, up to half of the cortex is dominated by vision. We are very visual animals. And seeing is believing. It's part of our language. Now, if you were a dog, what you would say if you could talk is that smelling is believing. Because they're much better at smelling. They have many more receptors, olfactory receptors, than we have. Now, here is one of the secrets, I think, of evolution, which was once you have parcellated the cortex and to topographically organize areas, then you can start stacking them. So here's, for example, the retina, the thalamic input to the cortex, primary visual cortex, and we're just getting started here, right? That's all that we saw in some of the primitive animals. And here's V2, V3, V4, infratemporal. There's 24 visual areas that have different functions, and information is flowing back and forth between them. And so this is one of the ways, again, it's like instead of gene duplication, you do duplication of areas. And we'll see later in the lecture that what consequences it has. If you have a deeper network, what does that buy you? What is the advantage of having so many cortical areas in a hierarchy like this? And we know that because we can actually begin to build these things, and we can see what functions they have. Now, this is a story that helps us understand how it is that we can have very similar animals with very different behaviors. How is it that evolution could create these different behaviors when 99.9% of the genome is identical? So this is a study of the mating behavior of 2 different species of vole. The montane vole that lives in the mountain and is very promiscuous, it doesn't settle down, and the prairie vole down on the farm, mates for life, monogamous. Well, let's look into the genome to see where the differences are. And it turns out that one of in terms of the change in both the maternal behavior and the paternal behavior really can be traced to a particular gene that is responsible for a vasopressin receptor. And it's shown here. This is immunostaining, so you can see that it's in the lateral septum here, diagonal band of Broca here, and it looks like it's being regulated differentially in different brain areas. And interestingly, if you look at the difference in the genomes of these 2 species, it's primarily here in the promoter sequence. The prairie vole has a longer repeat here at the beginning of the gene. And in fact, there's a variability in the number of repeats, and in fact, that's related to the degree to which the prairie vole will remain with, in a monogamous relationship. Now, we have these receptors too, right? They're found in all mammals. And it's interesting that humans have such a variety of mating behaviors, right? Some of us are very loyal to our spouse, and there are some that aren't. By the way, I should say the issue here is the male genes, right? And the experiment hasn't been done yet, but the question is whether if you looked at the size of the repeat in humans, you might be able to predict who it is that you're going to marry in terms of whether they're going to be faithful or not. This seems to be at least as important as knowing if you have a disease gene. Language. One of the most remarkable things is that humans developed language in a very, very rapidly, just a few 100,000 years ago. And we don't even know. It could have happened as early as maybe even 100,000 years ago. And the question is, what changed inside of the brain that allowed this to happen? It's a species-specific innate behavior. It's much more complicated, as linguists tell us, than simple calls. Although there are species like the songbird that can produce a whole sequence of syllables that has to be learned. And in fact involves the very same brain areas that we know are important for babies as they learn to speak. Very rapid changes occurring in the muscles in the orofacial area. Well, this turned out to be one of the genes that may be related to that shift from being able to call and have very simple signaling to very complex sequences. And this is a family that was discovered in England, and it happened to be a gene, FOXP2, a transcription factor, that actually shows up in all the areas that are important for regulating repetitive sequences. And in this family, each one of the black here indicate an individual that has a mutation in a gene, FOXP2, and also has a serious speech deficit. And I'm going to play for you an interview with one of the members of the family.
Where do you live, Laura? And how old are you? Where do you live? And how old are you?
So what you can hear is that this young child was struggling just to pronounce. If you listen to it, you can actually decipher some of it. But it clearly, the child knows what the right word is, just can't say it. And it could be that this is what we're seeing here is something emerging. And now let's look at the FOXP2 gene as it evolved over the primates. So each of these blue marks here is a change in the gene that's synonymous, which means that you get the same amino acid. And the red ones are non-synonymous, which means that it could change the function. And you can see that over 200 million years, the mouse, very little, one gene here that really had any impact. But in the human line here, there's two that within a very short period of time. And this really could be the change. And of course, the chimp here has a straight line without any changes. So this is clearly a potential gene, and it's a gene that shows up in all the areas that are known to be important for motor control. For example, the cerebellum here, and the basal ganglia, and the cortex and the striatum. And these are all very critical places. Now, everybody has been looking for more genes, and they haven't been found yet. There's got to be other genes that are important for development of language emergence in humans. But it could be that we're looking in the wrong place. So I was at a meeting with Sidney Brenner at Rockefeller, and he offered another hypothesis. He said everybody's looking for a gene that will create language, but they're not looking for a language suppressor gene that may have been turned off in the human lineage. And the chimp wisely left it on. And very, very acute point of view because of the fact that, in addition to language, in addition to giving us the ability to communicate, also gives us the ability to lie to each other. And the bigger we've gone from having small groups of people like here to radio, to television, to internet, and now language, really fake news, really is a problem because of the fact that an individual can reach so many people so quickly. Okay, now that brings us to this real issue, is what really makes us different, what makes humans humans. Now, the obvious thing is we have a much bigger brain. This is the chimpanzee, and this is our brain. Our DNA is 98.6% the same, so that means that those few million changes must be important. Now, we weren't the only creatures around. There's a whole line of Homos that disappeared, and in particular the Neanderthals that split off about 400,000. They actually left Africa 400,000 years ago and inhabited the western part of Europe, and then there's the Denisovans that left about the same time and inhabited the right east part of Asia. And we actually now have their DNA sequence, and lo and behold, it turns out that not only, and humans, by the way, they came out of Africa about 100,000 years ago, so we overlapped with these two other fellow species. And not only did we coexist with them, we cohabited with them. Because it looks as if like 4%, if you live in Europe, 4% of your DNA can be traced to Neanderthal origin. And if you're in the east part of Asia, 6% of your DNA is Denisovan. And it's in particular pockets. It's not just average. It's in particular the immune system has a very high percentage, the HLA locus, which may prefer some advantages, for example, to survive in a part of the world that we originally didn't come from. And so this leads me to assess, of all the differences, which are the most important? And everybody, if you ever ask anybody, what's different between us and chimps, they'll say, we have language. But as I just said, I actually think that languages has pluses and minuses, and I don't think we would have gotten here with just language. In fact, there's a real problem with transmitting information, because if one generation has to teach the next generation, they have to have everything in their head. And there's a limit to how much you can remember and carry on to the next generation, and especially if a lot of it has to do with myth, and a lot of it has to do with beliefs that really don't correspond to reality, which is what most early societies, of course, thought was the most important. But look, humans have extraordinary sociality. It used to be that if you go to a chimp troop or a macaque troop especially, it's really kinship, who becat whom, that determines who you are going to fight with or fight against. And in humans, it's amazing. We're willing to be social with almost anybody with a common interest, right? There are people who get together at chess clubs where it doesn't matter where you came from, it doesn't matter who you are. If you can play chess, you're welcome. And it's the same thing with sports. This is really something that humans are able to form associations based on abstract things, or even ideologies, right, which have even less concrete origin. Another interesting thing I discovered from reading the literature, and I was interested in education, it turns out that chimps don't have a way to train the young ones. It's by imitation. However, humans have something called joint attention, and it's where a mother can give something to the child, and the child then grabs it, and they both are attending to the same object. And once you can do that, you can then show the infant and the child how to do things, because of the fact that you're educating them about the world. And that then, in humans, had been expanded to a formal education system where we spent the first 20 years of our life sitting in a classroom with our heads having been filled with all kinds of facts and knowledge, right? That's remarkable. We've automated education. And that's allowed us to do a lot more than other species that just had a simple language. Now, what I think are the top two candidates for being the key to our advance and creating a civilization are first our amazing ability to learn. We'll be hearing more about that in Atsushi's lecture next. And this learning is much faster in humans than any other species. It has a higher capacity and it's much more diverse. And we're talking about learning of things in the world, motor learning, sports. We're talking about social learning. We're talking about all of the things that we've created, that we've learned how to manipulate. Tools, it's the obvious one, but there's, we've created computers, we've created all sorts of devices for being able to organize our existence around us, the buildings and so forth. And now finally, I'm coming to the one unique thing about humans that I think made civilization possible, and that's writing. So this is a cuneiform that goes back to 5,000 years. So we're talking now about something that is only a few thousand years old. Now, why was that so important? Well, it meant that you could transmit things that were learned in one generation to the next without being corrupted. And you can expand it because you can write a lot more than you could remember, right? And even more importantly, you can make corrections. So as you learn something new, you could erase what you thought the last generation knew, and you can add in and then build up a whole knowledge base that led to science and eventually led to our current civilization. So I really think that, and by the way, writing is not innate. That's why you have to go to school for 10 years to learn how to read, right? It's mapping language into your visual system. That requires a lot of training. But that's because we can learn. We have the ability to learn, which means that these last two are really very closely tied to each other. Now let's take a closer look at our brain. This is a pygmy shrew, which has one of the smallest mammalian brains, and this is in roughly the same scale, our brain. Amazing that you could scale something up that much by 5 orders of magnitude and have it still work the same way. I mean, what else do you know that has scaled up like that? Airplanes don't scale. It's because the laws of physics prevent you from just doubling everything. Even big animals have to run out of steam at a certain point. But there's a regularity, and this is a really classic regularity between body weight, as you can see here, and the brain weight. And these are all the species of mammals, except primates. You can see they fall in a straight line with a 2/3 power law slope of 2/3, and that suggests that what's important about the brain is not the volume of the body, but the surface area. And the sensorium is, in fact, proportional to the surface area of your skin, your eyes, and your ear. And what's interesting is that the primates, however, fall along a line that's above that, which means we have a bigger brain on the whole for a given weight than other mammals. And here's humans. Humans are yet above that, which means that we have an additional boost in terms of brain size. So this is important. It has more capacity. But what's interesting is that right next to us here is a whole other clade here, the porpoises and dolphins, which also are very intelligent creatures. And this is really fascinating. We're living with these creatures and really probably very intelligent, but we just don't speak the same language, right? So we have to try to understand a little bit about their form of intelligence. Now, even if you look at the primates, this is from a book by John Ullman, by the way, who's a visual physiologist, but has done a lot of work on primates. And it really illustrates something about your lifestyle and how important it is for your brain. These are all, each of these little icons is a different species of primate, and the ones that are leaf eaters are shown here. And you'll notice that they are below this line here, which is the average line for primates. Well, it turns out that leaf eaters, like gorillas, have put their resources not into their brain, but into their stomachs, into their digestive tract, because they have to eat a lot of leaves in order to get enough energy out of them. And interestingly, the insect eaters down here and the fruit eaters tend to be on the line. And again, here's humans, way above. Now, there's another feature that happens as we scale up a brain. It has to do with the ratio of the gray matter to the white matter. So the gray matter here is the outer 2 or 3 millimeters that, as you can see, is stained here in blue. And then coupling them are connections, axons that carry information back and forth throughout the cortical mantle. And that is shown here and is called the white matter. And what I've, back in 2000, Ketch and Zhang in my lab published a paper. We got a hold of volumes of the gray matter and white matter and just on a log-log plot, put them down. Here's the pygmy shrew that you saw earlier, and here's an elephant, and here's the human. I can't actually read it very well, but I think it's up there somewhere. Yeah, there we are. Dead on the line. In fact, this blew me away because as you can see here, the correlation coefficient, how well it fits those points, is 0.998. Do you know anything in biology that has, two out of 1,000 in terms of accuracy, right? There's got to be a law here somewhere. And we've thought a lot about it, but it really is telling us that the number of wires that you have is really highly regulated. And interestingly, the slope here is 1.23, which means that the bigger your brain, the more the white matter you have relative to the gray. And in humans, it's about 1 to 1. And here is mice. Oops, where's the mouse? Here it is. It's about 10%, it turns out, is white matter. So this is going to be very important for understanding communication between cortical areas, which of course is something that is very important for how the brain is able to globally organize information and not just have specialized areas for special domains. Now I'm going to come back now to our methylation project, and now this is a paper that's been recently accepted in Science. We took it from looking at the average methylation pattern in all neurons and all glia, we compared the two, to being able to bring it down to single cells. So we have single cells, we're able to tag them, and then we're able to do the methylation pattern for single cells, and we had about 4,000 cells, and they clustered according to this diagram. The red ones are the inhibitory cells, and you can see there's about there's about 1, 2, 3, 4, 5, 6, 7, 8, 9 different ones here. But it turns out there's even more different pyramidal cells, and it depends on the layer the pyramidal cells are in. This is human and layer 2, 3. This is human layer 5. Look, there's 1, 2, 3, 4 different clusters. And now in 2 dimensions, you can actually be able to distinguish where they live. These are all the inhibitory cells on the right. These are all the pyramidal cells. Layer 2, 3 is at the top. Layer 6 and 5 deep layers are at the bottom. I don't know why it worked out that beautifully. And what we discovered in comparing them to mice is that it looks as if humans have a couple of new cell types that aren't found in the mouse, and more differentiated pyramidal cells. So we're beginning to see here at the level of fine grain in terms of the different cell types, what has really evolved in the human cortex. Now finally, what I want to do is to come back to this idea of a hierarchy, because it's been only a few years since we've been able to develop artificial neural networks that have the same deep hierarchy. Back in the 80s when I was working on neural network learning algorithms, we had one layer of what we called hidden units between the inputs and the output. And that really got us off the ground, but it really wasn't very powerful. It was very difficult to train them up because you needed a lot of data. And the computer power back then was puny compared to today's. Well, fast forward, computers are 1,000,000 times faster. With the internet, we have 1,000,000 times more data. And we can have many more units, many more layers. And this is now, it's called a deep learning convolutional network. It roughly mirrors the visual system that's seen in the primate, in the sense that the input from the image, for example, gets processed and then represented by a bunch of features in V1. And these features now are convolved across the image because they're the same. And in the case of V1, we know there are these simple cells that are oriented, and those are, in fact, ones that are going to be present in our deep learning network, but there are going to be many more layers. And what's interesting is that the transformation, the nonlinear transformation between the layers, that proved to be the most effective for training networks to recognize objects in images turn out to have the same kind of nonlinearity you see in neurons, a threshold nonlinearity, a linear filter, threshold nonlinearity, pooling, which is called a complex L in V1, and then normalizing so that's within the same dynamic range. And you repeat that over and over again. The more layers, the better. And at the very end, you have an all-to-all network, which then is able to recognize objects. And there was a contest called ImageNet, consisted of a database of roughly 15 million images and 20,000 categories. So this is a big data set. And this was back in 2013 when it was announced that the recognition capacity of this architecture, at a NIPS meeting, by the way, right, had reduced the error rate by 20% in terms of recognizing which category an image was in. And it doesn't, 20%, you may not think that's a lot, but the typical improvement that occurs through the traditional engineering method by engineering the features for each object that are going to be representative and discriminative would typically be 0.5% per year. So literally it amounts to a 40-year jump into the future. And it has important consequences because this is behind the new AI that you've been reading about. Because this can be used not just for image recognition, it can be used for natural language, it can be used for speech recognition, and it really has improved performance in every one of those areas. Here's a really amazing demonstration. This is an image of a cluttered scene, and you can use this deep learning network to recognize all the different objects, and then put it through a recurrent network. The one I've showed you just now, the convolutional network is feed-forward, but we know within the brain there are recurrent connections that allow information to be circulated, and that allows you to do things like recognize sequences. And indeed, it was trained up to take in all the objects and then to put out a string of words that represented a summary or a caption of that scene. So a group of people shopping at an outdoor market, which is not bad, right? I mean, that's probably something that you might have come up with. This one's a little dubious. There are many vegetables at the fruit stand. I'm not sure. You have to look very carefully what's actually there, but it could be that there's a mixture. Here's a couple more. In this particular case, the network, recurrent network, didn't just give you a sequence of words. It actually highlighted for a given word the part of the image that was responsible for that word. So here's a woman is throwing a frisbee in a park, and here's the frisbee. A dog is standing on a hardwood floor, and here's the dog. I'm not sure if it's standing, but it's there. A little girl sitting on a bed with a teddy bear. Well, there's a teddy bear, I'm not sure it's a bed. A group of people sitting on a boat in the water, and there they are. This is amazing. This is natural language. We're not talking, this is the last thing I would have expected 30 years ago when we were starting, that this would be so effective at things that humans do very easily, but it turned out that AI, traditional classical AI, was basically failed at. So we're living in a new era. But that's not all. There's more to the brain than the cortex. And so another part of the brain that's extremely important is called the basal ganglia. It's below the cortex. It gets input from the cortex. And it's responsible for you learning sequences of actions toward a goal. And there's a theory that there's a key learning algorithm that's called the temporal difference learning algorithm for reinforcement learning. So this is classical conditioning, but you can do that for a sequence of actions. And the theory, which was developed back in my lab in the 90s, was that the dopamine neurons that project very broadly throughout the cortical mantle and this basal ganglia shown here, these are the different areas that are getting input from the cortex, that they're computing reward prediction error, that they're telling you how good, from you predict how much reward you're going to get for an action, and it will tell you from the reward you get the error. Either you got more reward than you expected, increase the strengths, or if you got less reward than you expected, then decrease the strengths. So this is a very simple algorithm, but when you couple it with deep learning, you can get superhuman performance. And that was demonstrated in the game of Go, which is very popular here. so I don't think I have to explain how Go works. It's much more complicated than chess. The board is much bigger, 19 by 19. The number of possible game positions is greater than the number of atoms in the universe, and it's a very high art here in Singapore, China, Japan. Now, back a year ago, this is March 2016, AlphaGo, which was developed by DeepMind, which is a company in London that was founded by a neuroscientist, used temporal difference learning coupled with deep learning to play Go at a very high level. In fact, at this point, it beat everybody in the company, and they had no idea how good it was. In fact, it had beaten the European champion. But the level of play here is much higher. So EC Dahl was the South Korean champion, 8th-time world champion, and clearly he is having, look, you can see he's really struggling here. And he lost the first 3 games in a 5-game match and was absolutely, totally devastated. And at a press conference, here's a quote: "I should have shown a better result, a better outcome, and better content in terms of the game played. And I do apologize for not being able to satisfy a lot of people's expectations. I kind of felt powerless." The AlphaGo made moves that no human had ever made before, and subsequently on analysis have shown to be breakthroughs in terms of whole new game positions that are really very powerful. So AlphaGo is actually creative. This is not something that was engineered in. It learned by playing itself hundreds of millions of games. Now, EC Dahl is not the world champion. He was beaten 18 out of 20 times by Ji. And by the way, these Go champions are like rock stars, right? They're like, Muhammad Ali was in the US in boxing. A public figure that everybody knows and respects, right? Because there are 2,000 Go professionals in China, for example. And this is after the loss. EC Dahl won the 4th match, saving face for humans, and eventually lost 4 out of 5. But Ji said in response, "AlphaGo can't beat me. He's the alpha male." And here's what happened. A year later, they had another match in China, and it got a huge amount of publicity. Every TV station was carrying these matches in real time. And, it was that much interest. And so here's what happened. In a 3-game match, he lost all 3. And by a big margin. There was one match where he only lost by a few percentage points. But that broke him. That was the first match. And he was playing well up to that point. January 5th. 2017. After humanity spent thousands of years improving our tactics, computers tell us that humans are completely wrong. And in another statement, he said, "AlphaGo is the god of Go." It's superhuman in a very narrow domain, but it's built with a very simplified version of the architecture of the brain, and just 2 parts out of hundreds. So there's so much more that the brain needs to do in order to be autonomous and intelligent. And this has now gotten an enormous amount of attention in the press. And the NIPS meeting that I've been running now for the last 25 years has been increasing in attendance. It used to be around 10 years ago, about 600 to 800 people. But the last meeting, there were 6,000 people in Barcelona last December. So and we had like 60 major companies there that were recruiting, and all the famous ones, Google, Microsoft, Facebook, and so forth, including financial firms, including driverless car firms. It really was truly remarkable. But this is not the most exciting thing that's happening. I think it's important, and it has a lot of financial impact. But what's really interesting to me is that we're going to have a similar breakthrough in understanding the brain over the next 10 years. And it's already happening. And I'll just give you a little taste. This was announced in the White House on April 2nd. This is President Obama's signature science and technology project, right? The grand challenge, called the BRAIN Initiative. And it's an acronym, and the last 2 letters, I and N, tell you everything. Innovative Neurotechnologies. Get neuroscientists to collaborate with engineers. with people who know how to build things. And if the neuroscientist knows the question and needs to have a technique or a tool to be able to answer it, the engineer knows how to build tools, and if they two can just get together, magic might happen. And it has. In the last 3 years, there's been an efflorescence from the NIH in terms of funding these teams and the results that are coming out. I was on a committee which was commissioned by Francis Collins, the director of NIH, and we were charged with trying to come up with a plan for the next 10 years to advance neuroscience. Which are the areas that have the highest priority? Which are the questions? Which are the issues? Milestones, budgets. It took us more than a year, a group of 15 people led by Bill Newsom at Stanford and Corey Bartman at Rockefeller. And just getting 15 neuroscience degree about anything, by the way, is an achievement, because everybody, of course, thinks that what they do is more important. But we did it, and it was adopted, and it's the first time I've ever written a $5 billion grant proposal for 10 years. And by the way, it funded the single cell work that I was showing you about identifying single neurons and figuring out what their identity is. But more importantly, it's now possible to record simultaneously from 80,000 neurons. And I'm going to end on this just to give you a feeling for where we are. Right? Most of the time that I was a neuroscientist, it was using the single microelectrode technique. It was possible to record from one neuron at a time. And with 86 billion neurons in the brain, that's going to take you a while. But it's worse. It's worse. Although we have a pretty good map of how the neurons respond, imagine trying to figure out what's in a room if you have to look through a soda straw, one pixel at a time. Right? That's what we've been doing for the last 50 years. And that brain doesn't work one neuron at a time. The brain works through interaction between millions of neurons all over the brain, right? So we now have tools and techniques. This is a calcium indicator, GCAMP, which is in a zebrafish brain. Zebrafish is actually fixed in a dark room so that there's no sensory input, no motor output. So this is really what's called spontaneous activity. Each of these little dots here is a neuron, and it flashes when it's activated, when it has a lot of action potentials, and calcium comes in. This is from the top, from the front, and from the side, and this is taken with something called a light sheet, so it can go up and down 10 times a second, and so it can scan the whole volume of the brain. You're just looking at one cross-section here. So just look at the pattern, just see what's going on. In fact, there's information going up and down the spinal cord, and there's activity here in the forebrain, and this is the eye. And you can see things flashing, and you get a sudden activity pattern that actually spreads. This is called the Chinese curse. May you get what you wish. We've been asking for this for a long time, and now we can see it, and we have no idea how to interpret it. No idea, because of the fact that it's just foreign to us that you can have so much activity going on when there's ostensibly nothing happening outside. It shouldn't surprise us, because in fact, when you're sitting in your bed at night and things are happening in your head, thoughts going through and so forth, it's got to be activity in the neurons, right? Patterns of activity floating back and forth. But now we have access to it. We can eavesdrop on the brain. This picture was taken literally 10 minutes before the announcement was made, and the chief agencies and representatives, Francis Collins, for example, from NIH. This is Cora Merritt from NSF. Now this woman here, I was talking to her, and I had never met her before. I had no idea who she was, and so we're talking about what's coming ahead, because why was she there? And she said, I'm the head of DARPA, the Defense Advanced Research Project Administration, and we're going to be working on humans. And indeed, they're putting electrodes into humans, not just to record, but to stimulate, and they want to close the loop. And these are cowboys, let me believe you. They really they're just charging way ahead of us, but we'll see. But in any case, as I was talking to her, suddenly there was a tap on my shoulder. I turned around, and there he was, Obama, towering over me, much taller than you see in the pictures. Or either that or I'm much shorter. And I was so flustered, I hadn't prepared anything, and all I could say was, "Thank you, Mr. President, and thank you."